Hi everyone, myself Naveen, finally a medical student, and in the short video session we'll be learning about complete information about sodium potassium pump, and mainly conceptually understanding how many sodium or potassium is being transported across the cell and which ion is being exported outside and which comes towards inside. Few important and fascinating points about sodium potassium pump is that it is ubiquitous. It is universally present in all the cells in our body, and it is an electrogenic pump. That is, it contributes for maintenance of resting membrane potential, which we'll be learning in few minutes in this session, and it is also the one which creates ionic difference between the extracellular and intracellular component. Next one is that it is heterodimer and heterogeneous. That is, sodium potassium ATPase pump as such has two components, that is alpha subunit and beta subunit, and there are different subunits, or even in that alpha and beta subunit there are heterogeneosity, that is alpha 1, alpha 2 and alpha 3. These quite very found in different places, and beta 1, beta 2, beta 3 as are seen in different kind of organs and tissues. This is the main theme of this session, that is the coupling ratio. What is the coupling ratio and why it is 3:2? And if you see the location of the sodium potassium ATPase pump is mostly around 95% plus it is basolateral, that is it is present near a blood vessel. This will be understood in the upcoming few minutes, and there are some places it is found in apical regions also. If you know, comment in the comment section. Two important things which we already know, that is resting membrane potential of any cell. You know, every cell has a resting membrane potential is always negative. For example, minus 70 millivolts seen in nerves, and minus 90 seen in cardiac ventricles and skeletal muscles, like that. We know resting membrane potential is negative. This negative can be achieved by two ways. That is, negative, what that it means is that negative is present intracellularly compared to the extracellular component. That can be achieved by two ways, that is gaining an electronegative ion. For example, chloride, from extracellular to intracellular we'll get electrically it is more negative, right? We'll get electric charge that is negative inside the cell. Or losing a positive charge from inside to outside brings about the same response. Next is active transport. The main concept of active transport is that we use an energy molecule, for example mostly ATP, and that energy is being used to move a molecule against the concentration gradient. Now, quick bite is that we know sodium is the predominant ion seen in extracellular fluid, extracellular compartment, and potassium is predominant ion of the intracellular compartment. So one thing is that we use ATPase, that is energy, in the sodium ATPase, potassium ATPase pump. So we are using energy. What does that mean? We have to move an ion that is against the concentration gradient. We know the intracellular is potassium predominant, extracellular sodium predominant. So what do? We use this energy and move in opposite direction. Got it? This clears the point, the direction of movement of ions. So sodium is predominant outside. We are moving sodium against the gradient, that is sodium is more outside, less inside. Normally the tendency would be to move from outside to inside, but we are using this energy to move the molecule from inside to outside against the gradient, and same way for the potassium, which is low outside, high inside. that is the reason we are using ATP and hydrolysis being happening in the alpha subunit. With this energy we are moving against the concentration gradient. This is the first part. Next is that RMP is negative. If we see, one of the important functions of sodium potassium pump is contribution of resting membrane potential. Though it is less, but it is important, right? It contributes around -4 millivolts of electronegativity for the resting membrane potential. What did I say? To gain negative, we should gain an electronegative ion, or we should lose a electropositive ion. In this case, both are positive. So the net amount of movement, that is, you lose more rather than you get. That is the reason if you move 3 sodium and get 2 potassium, the net gain is -1. That is, you are losing 3 positive ion, rather you are getting only 2 positive ion. Both are equivalent, is +1. The charge is +1, right? So the net gain is -1. So now we will get to know the quantity of sodium is being pushed out is 3, the gain of potassium is 2, and their direction is also being discussed earlier. Now the final thing, the functions. Sodium is being moved out. Sodium being osmotically very active. Sodium and water are like besties or boyfriend and girlfriend. Anyway, wherever sodium moves, it drags the water along with it. That's what a professor used to say. Wherever sodium moves, water is being dragged towards it. So it is regulating the volume of the intracellular component. If more the activity of sodium potassium pump, more is the sodium being pumped out, more is the water lost. So ICF would be less in water content, and vice versa. Already discussed -4 millivolt contribution to the resting membrane potential, and sodium potassium pump is the principal or classical example of active transport, primary active transport. Because of this, there is an electrical or electrochemical gradient that is being formed. For example, potassium is more inside, 3 sodium is being pushed out, so external is sodium predominant. Like this, electrochemical gradient is being used for secondary active transport. For example, S-glut, sodium and glucose co-transporter. This is an for example of secondary active transport, that is co-transport. Sodium and glucose moves together. See here, sodium potassium pumps out sodium, and that sodium will be used for transport of glucose along from external to internal towards ICF. I want you to comment what are the types of S-glut transporters found and where are they found. It's quite easy. Comment in the section. And another sodium potassium 2 chloride pump in thick ascending loop of Henle, that is also being functional only because of sodium potassium pump. Another one thing, there are alpha is being the main active component of sodium potassium pump. All the binding sites, sodium binding site, potassium binding site, ATPase hydrolysis site, is all found in alpha unit. Another most widely used drug, let's say for example congestive cardiac failure, even that has this activity, sodium potassium pump activity. Just comment that in section. And another similar to that drug directly acts on the sodium potassium pump and inhibits the activity of this pump. That's the binding site seen in extracellular surface of the sodium potassium pump in alpha subunit. Just remember that also. And at last, hypothyroidism being the most common preventable cause of intellectual disability in a newborn. Even that thyroid hormone increases the basal metabolic rate, or that is done by activating increasing the activity of sodium potassium pump and thereby increasing basal metabolic rate. And this also has activity in increasing neuronal development and neuronal maturation. So you can link that low thyroid, low sodium potassium pump activity, reduced brain development and maturation. So it is the most common cause of preventable intellectual disability in a newborn. Thank you. Have a nice day. If you like the video, just give a thumbs up and subscribe to the channel. We'll see more such videos in the upcoming times. Thank you.